In this presentation, I will discuss with you the basics of cell culture that we will be using in our lab. One important aspect about cell culture is the type of cell culture that one is doing. And there are essentially two types: primary cell culture or mortalized cell lines. In primary cell culture, one obtains the cells that one wants to culture directly from the animal or the organism. One takes those cells out of the animal, places them in culture, and then will have them grow. One of the issues about primary cell culture is that eventually the cells will undergo senescence. In other words, they will start displaying old age, often related to telomere length, and then they'll start dying or misbehaving or becoming abnormal. And so that's a disadvantage of primary cell culture, but the big advantage is that you know what types of cells they are because you isolate it from the animal. Another type of cell culture is immortalized cell lines. And these are cell lines that don't undergo senescence, and they keep growing and growing and growing. And why? Because they are like cancer cell lines. So they've either been isolated from tumors, or they've been primary cells that have undergone mutations that make them more like a cancer cell. And so these immortalized cell lines, you can keep growing and growing and growing forever, which is great, but then they have a certain amount of abnormalities because they're like a cancer cell, and so one is never certain that they're exactly like the cells in the body. So when performing cell culture, one tries to have conditions that will be very similar to the in vivo or the conditions that the cells grow in normally. And so some of these factors include temperature. Cells are usually cultured at body temperature, which is 37 degrees Celsius, and that is the optimal temperature for most human or mammalian cells. Cells normally are in the body enclosed within skin, and there's a fair amount of moisture, and so one tries to keep the humidity there so that the cells will be basically in a fluid environment and the fluid will not evaporate. Normally cells are growing in a fluid that is pH balanced, in other words buffered. And so again, when we culture cells, we try to keep the pH in a normal range of about pH 7.2 to 7.4. Importantly also, cells need to be in the right salt solution, because if it's too low, they will start absorbing water and may actually pop, or if they're too high, they'll shrivel up. So you want a osmolarity or salt solution surrounding the cells to be isotonic, so the cells would grow naturally. In addition, cells need oxygen for metabolism, and so you'll want them to receive air containing a certain amount of oxygen that would provide the appropriate amount of oxygen for aerobic respiration. And finally, very few cells grow just in a solution. Most cells, like cells in your body, need to be somehow associated with a tissue. And usually must be some sort of substrate or material that will mimic the normal extracellular matrix environment that the cells grow on in the organism. And so for this, we use specially treated cell culture dishes that have a treatment on there that does mimic this extracellular matrix. Unlike regular petri dishes that are used for bacterial work that don't have this substrate, and the cells will not attach. So one big issue with cell culture is avoiding contamination, specifically with bacteria or other microorganisms. And the way to think about this is, imagine, as I say, you got a dish of cells. Mammalian cells, most cells that we grow in culture, will double about every 24 hours. On the other hand, bacteria, if you got bacterial contamination, bacteria usually double on high nutrient medium about every 30 minutes or so. So then think about the situation. What if you started off with 2 million animal cells, but one bacterium got in there? Let's think about what would happen after 2 days, 48 hours. Think through the math. For your mammalian cells, they double every 24 hours. So 48 hours is 2 doubling times, so 2 to the 2, or 4 times the number of cells, 2 times 10 to the 6. That means after 2 days, 48 hours, you would have 8 million cells. That's quite a few cells. But let's think about what would happen with a bacteria. You just have one in there. But it doubles every 30 minutes. So in other words, 2 doublings an hour, 48 hours times 2, so 2 to the 48 times 2, or 2 to the 96. That would be 8 times 10 to the 28th bacteria. And so you'd have significantly more bacteria than you would animal cells or mammalian cells after 48 hours. And so that's the issue with contamination. If you get it contaminated with bacteria or something else, essentially the bacterial cells will take over the culture and your cells would eventually die. So for growing cells, you need to have them in an appropriate cell culture media that would have all the necessary ingredients for those cells to grow. And there's a variety of different cell culture media. One of the common ones that we're going to use is called the Bulkos Modified Eagles Medium, also known as DMEM. And so this culture medium will provide all the essential nutrients for cells to grow. So what are some of those important nutrients? So this is the formulation for the DMEM media. And there's a variety of different things that are in that are important for cells to grow. And I want you to have a good understanding of some of the major aspects of it. For instance, there's a variety of inorganic salts present. And one of the purposes of these salts is to keep the media isotonic and maintain the osmolarity so that the salt or solute concentration matches the normal body concentration. In addition, in this salts are various minerals that the cells will need. Another nutrient that is provided for the cells are the various amino acids. Most mammalian cells cannot produce amino acids naturally, and so we supplement the media with them. In addition, you'll notice that L-glutamine there has a much higher concentration than the other amino acids. And that's because the glutamine can be used as a nitrogen source to provide nitrogen, in other words, amino groups, for various other metabolic processes. In addition to amino acids, there's a variety of vitamins that are provided for the cells that they need. And then finally, you'll notice down in the other side a couple of things. Provide glucose. Glucose is basically sugar and energy source for the cells, and that's why the cells will use the glucose. In addition, there's something called sodium pyruvate, and it is used to help feed the citric acid cycle for basically metabolism in the cells. In addition, I want to point out 2 other things that you will notice in the cell culture. One is phenol red. I'll come back to that in a minute. And the other is sodium bicarbonate, NaHCO3. And essentially those are involved in pH balance. So the buffer that is used to maintain the pH of the cells in culture is a bicarbonate carbonic acid buffer that is essentially similar to the buffering that is done in your blood. And so essentially included in the cell culture media is sodium bicarbonate, which release bicarbonate ions, And those bicarbonate ions will absorb any excess H+ ions to help keep it buffering, and then it will go to H2CO3. To provide carbonic acid, we add 5% CO2 to the incubator. That's released in the air. That CO2 will mix with water to form carbonic acid. That carbonic acid will dissociate to bicarbonate ions and H+ ions. So if there is too few H+ ions, the carbonic acid can supply them. And so we have this buffering between the bicarbonate ion and the CO2 that forms carbonic acid that keeps the pH in the cell culture media about 7.2 to 7.5 or 7.4. The other thing that I want to mention, you'll notice that the media is a little bit reddish orange, and that is due to the addition of phenol red. Phenol red is a pH indicator. Under normal pH of about 7.2 to 7.4, it's this orangish red color, and that indicates that the pH of the media is correct. If the pH becomes acidic, the phenol red will turn yellowish, and so then you could tell the media becomes acidic. If the media becomes more basic, more alkaline, the phenol red will turn a magenta color, and so that again will tell you that the pH is off. Why might the pH go off? One thing is, if you don't have CO2, you'll have an excess of bicarbonate ions. If there's an excess of bicarbonate ions, it's going to make it slightly more basic. The other thing is, if you get contamination, the bacterial cells will start using up the nutrients and releasing waste materials, often lactic acid, and so that will cause the media to be more acidic. In addition, if you have the cells growing a lot and they're using up all the nutrients and releasing waste products such as lactic acid, the media again might turn acidic. In addition to the basic nutrients that are present in DMEM, sometimes the media is supplemented with various things. One thing that it is, again, supplemented is the phenol red. The other thing is something called FBS, or fetal bovine serum. And this is essentially blood, after the blood cells have clotted, from a fetal cow. And why do we use this? Essentially, this fetal bovine serum has many different nutrients in there that are helpful for cells, including many growth factors that are involved in fetal growth. And these growth factors are actually usually very helpful for cells. And so if you don't want to specifically add all these growth factors, or you don't even know what growth factors specifically help your cells, the addition of fetal bovine serum, FBS, usually around 10%, will have all of these things in it. They'll help your cells to grow. And then sometimes you can add other supplements to the media. So now I want to talk about a very important part of cell culture, and that's aseptic technique, or how to keep the cells from being contaminated. And so there's a variety of things that are involved. One of the most important things is we do all our work in a biosafety cabinet, often called a laminar flow hood. And it's very different than a fume hood that you use with chemicals, because the idea with this one here is it protects both the inside, where you're culturing the cells, and the user from potential contamination going in or out. And that works because in the fume hood, air is just taken out. But in a biosafety cabinet, air is brought in, but it's not going directly onto the cells, but it gets sucked in and circulates around, so that there is a set of circulating air going around that forms this air curtain. And that air curtain, which you sort of see by the blue arrows here, will actually push and prevent bacteria that might be coming in. They would get sucked into the system. There is up above a filter that will filter out bacteria. Likewise, if you're working with organisms that there's a biosafety concern, that anything that might come from them in the hood would get sucked up in this air current, and so would not then come out of the hood and infect the user. Other aspects that are used to keep this aseptic environment is that in the hood one sets up a shortwave UV to decontaminate it when it's not in use. One tries to use only materials that are sterile with the cell culture and only open them in the hood so that any contamination would be basically reduced. One uses 70% ethanol to decontaminate things, both spraying down the hood before as well as after use, as well as spraying down things that might go in. And then, in general, one wears gloves and usually a lab coat to protect yourself, but also to protect your cells. Other thing is when you set up your hood, you want to set it up so there'll be the minimal chance that as you're going from container to container situation, that you would accidentally drop something into your cell culture dish. So usually one puts the cell culture dish in the center, that's what it's working with, and then the various pipettes or media or other things you're working with around the outside, and so that you wouldn't naturally cross over and potentially decontaminate them. So now that you know some of the basics, I want to talk a little bit about the procedure we'll be doing, and that's what's called subculturing cells. And so essentially, whenever cells have used up the nutrients in the medium and gotten too crowded, we need to subculture them and basically put them in a new dish and give them more room to grow. And in doing so, we only will put some of them in the new dish so that there'll be more space to grow. And so this is the protocol, same as the one that I posted on Blackboard. And I want to point out a couple of things to do that. First, the cells are attached to the substrate, so we'll take off the old media. We'll wash the cells, and I'll come back a little bit to why we do that. And then the next thing is we need to get the cells off the substrate. In other words, get them off the dish they're attached to. And we use something called trypsin EDTA or trypsin versane for this procedure. And trypsin versane or trypsin EDTA contains two things. It contains EDTA. EDTA will chelate calcium and magnesium. And essentially cells have cell adhesion molecules. Some of them are calcium dependent. And so if you move the calcium from the media, those cell adhesion molecules will release. And so that will help release the cells. In addition, there are other cell adhesion molecules that are not calcium dependent. And so we include the enzyme trypsin. The enzyme trypsin is a protease and it will cut proteins. And so it'll cut these other cell attachment proteins and therefore help release the cells. Thing to keep in mind is trypsin is an enzyme, and so it will keep working. And so you don't want to incubate it for too long. So I say incubate for 5 minutes, because if you keep incubating, it's going to cut up all the cells in the cells, and then they're going to die. The other thing is you note that we place the flask in 37 degrees Celsius. And again, that's because the enzyme mediated reaction, like most reactions, will proceed faster at a higher temperature. And so at the 37 degrees, we'll proceed at a reasonable rate so that in 5 minutes we'll get the appropriate amount of cutting. So then after we treat with a trypsin EDTA for 5 minutes, we want to stop it because we want to stop the enzyme from cutting. and we stop it by the addition of complete growth medium that contains DMEM plus 10% FBS. And so there's a couple of things in this FBS that help in stopping the trypsin. One is that FBS has a bunch of other proteins, including bovine serum albumin. And so these other proteins act as a competitive inhibitor for the trypsin. The trypsin would attack these proteins instead of the cell proteins. The second thing is that the FBS actually has in it a component called trypsin inhibitor, a molecule that will actually inhibit the trypsin. And so the addition of this growth media will stop the trypsin from working, which makes me come back to the second step where we add PBS to wash the cells before we add the trypsin. And that's because, if you remember, our cells are growing in DMEM plus 10% FBS. So even though we take off the media initially, there still may be some FBS and some trypsin inhibitor present. And so before we add the trypsin, we want to get rid of that trypsin inhibitor, so we get rid of the PBS, or we get rid of it using PBS. So after adding the media, we want to take and squirt off the cells, wash them off the dish. We put them into a tube and we will gently centrifuge them, bring them down to the bottom. We'll remove the old media that contains some of this trypsin. Then we'll add some fresh media, pipette it up and down to resuspend the cells in the media, and then take a portion of those cells, either 10% or 20%, in other words 1 to 2 mils of those cells, add it to a new dish and add fresh media, 8 or 9 mils of new media, so that essentially they will start growing again, and we now have a new dish of cells that contain a smaller number of cells, and these cells can begin to grow and start repopulating the dish. So one of the things to think about is, why are we doing this subculturing? And so a variety of things. First of all, the nutrients in that media get used up. As the cells are growing, they're using up the nutrients, and we'll end up with less and less of them. Second of all, the cell releases wastes, and so we need to get rid of those wastes, as well as some of the dead cells. And so by subculturing, we get rid of those. And then finally, cells don't like to be too crowded. If they get too crowded, there's a phenomenon known as contact inhibition, and they just stop growing because they're contacting cells on other sides, and they're sort of squished in. So the other thing I want to talk about is a little bit about counting cells, and there is a device known as the Hemocytometer that we can use to count cells. And essentially, the Hemocytometer was originally developed for counting red blood cells. Gives a slightly different name, but it actually works as a very convenient way for counting mammalian cells as well. And one does it by putting cells on this grid pattern. And if you look in the center, there is a set of boxes outlined by the red circle. And that set of boxes, or any set of boxes that parallels it on any of those sides, is of a very specific size. Each side of that box is 1 millimeter, and when the cover slips on, there's a depth of 0.1 millimeter, which makes a volume of 10 to the 4th mils in that area. And so one can count the number of cells in that area, and that is the number of cells in 10 to the 4th mils of the media solution that you've added to this hemocytometer. And so essentially, after counting them, you'll take that count and multiply it by 10 to the 4th to get the number of cells per milliliter. One can also count multiple regions and average them, for instance, counting regions 1, 2, 3, and 4, and get an average, which will make it more accurate. And so as I finish up this presentation, I leave you to think about what are some of the things that could go wrong, what are some of the errors that one could happen to have, and what would be the results as you review the things we've talked about.